Hello and welcome to the 9th episode of the Innova Biosciences webinar series. Today's webinar will discuss Western blotting, the principles and a comparison of indirect versus direct immunodetection. Our speaker today is Dr. Brian Carpenter. Brian completed his PhD studying the molecular mechanisms of the WT1 tumor suppressor protein at the Center of Gene Regulation and Expression, the University of Dundee, which was followed by a postdoctoral position at the University of California in San Francisco. Throughout his scientific career, Brian performed and optimized a large number of Western blot experiments and became adept at troubleshooting and is keen to pass on his knowledge. During today's webinar, he will discuss key elements of the Western blotting technique along with helpful tips and hints. The subjects that will be discussed today include sample preparation, SDS-PAGE, Western blot transfer, blocking, indirect detection, and indirect versus direct antibody detection, direct detection for multiplex fluorescent Western blotting, problems with sourcing labeled antibodies for direct detection, which will be followed by explaining the simple solution, which is the antibody labeling kits, and questioning the secondary antibody amplification hypothesis.
Thank you, Claudina, and thank you everyone for joining us on for this webinar. First thing is I would like to set the scene of the webinar. When I started doing a bit of research on the web in regards to Western blotting, I've found some really useful webinars which have been developed by companies such as Abcam, Novus Biologics and Protein Tech, and I thought they really they nicely told us about the fundamentals of Western blotting, and therefore I wanted to try and understand how I could add value to these webinars. And therefore I thought about providing an informative webinar focusing on hints and tips to increase the chances of obtaining a successful Western blot based upon my own experiences. And it's my ultimate ambition is to create a checklist for yourselves to be able to assist in the troubleshooting issues you have and thus allow you to facilitate a successful Western blot. So I liken Western blot to actually building a car engine. Basically, without following the instructions, you do not know if the car will start or not until you have assembled all the parts and you turn the key. And we've all done this with Western blotting. We rush through the experiment. We don't put in place fundamental checkpoints, and what we end up here is a situation where we end with a blank blot or a black blot, and we have to try and understand what went wrong. And here's myself many years ago looking at these blots thinking, well, what has gone wrong? And then what transpires is I have to end up spending a lot of time and effort, and here I am burdened with all this work to try and end up with the perfect blot, and here's an intermediate. And it's based upon these experiences that I have found that allowed you to go from having a blank blot to having one which we all aim for, which is the perfect Western blot, is to put in place a fundamental checklist for yourselves to go through to assist in the troubleshooting process. So one thing as I would suggest is before you even pick up a Gilson or a pipette is to actually do some desk research to try and understand more about the protein or the target protein that you're interested in. You know, there's lots of information in the literature and various websites such as the Human Protein Atlas, which has a lot of useful information. Well, let's just start at the very beginning of the expression of the checklist. So, you know, one thing that's important is to determine where the protein is expressed. It's pointless running HeLa cell extract if the protein's more is expressed abundantly in 293 cells rather than in HeLa cells. And one thing that can be very useful in understanding is where in the cell the protein localizes. Is it in the nucleus? Is it in the membrane? Or is it in the cytoplasm itself? And therefore, that will, by understanding that, you can enrich your population of cellular lysates by isolating the various parts of the cell that the protein localizes in, whether that be the membrane, the nucleus, or the cytoplasm. And one thing that's worth noting is the post-translational modification can actually have determine how the protein migrates through the gel. For example, I worked on a protein called brain acid soluble protein 1, BASP1. is its abbreviation, and it was found to actually, it was predicted to migrate around 22 kilodaltons, but through to a post-translational process called myristylation, it actually migrated around the 50 kilodalton molecular weight bands. And therefore, you know, if I hadn't been aware of this at the time, it could have actually resulted in me taking what was a very nice Western blot and actually scrapping it and going back to the sort of design board to try and understand what was going on. And there's all of this can essentially be avoided by doing your desk research to thoroughly understand where your protein is. And then we move on to the extraction. You know, you have to design a sample extraction protocol based on the information you gained from the previous step. You know, it's important to choose the right buffer, and there's plenty of instructions out there in regards to what type of buffer you would use, whether it be Reaper buffer, or whether you would use Downs homogenization. The list is, the information on the websites are quite abundant. But again, this will affect on how the localization will affect how you design this step. You know, it's pointless isolating the membrane fraction if you find that your protein's isolated within the cytoplasm itself. And one thing is to eliminate your protein being degraded very quickly is to add the necessary protease inhibitors such as PMSF. You know, it's always important to make sure that these inhibitors are freshly prepared. One thing is, if you're studying kinases or phosphatases, again, once a cell is under stress when you lyse it or you're preparing it, it's important to have phosphatase inhibitors present which prevent the cell stress pathways being activated and then resulting in your protein not being modified in such a way which doesn't reflect it being under normal cellular conditions. And again, one of the things I would recommend is always to prepare your buffers fresh. I never once used any buffers unless it had been prepared fresh on that day, and also to have all your buffers cool and prepared. You know, it's pointless having them at room temperature and then putting the cells in this type of buffer, because as the temperature increases, the more likelihood of your protein or the proteins being degraded. And one thing is, I can't stress enough, it's very important that you clean all the tools, such as homogenizers, to ensure that there's no residual protein left over from previous preparations. And also you don't want to be contaminated through being having residual proteins left over. And again, it's important to have all your samples, to prepare your samples quickly and efficiently. And what I mean by this is have all your buffers clean and prepared, fresh, sitting there on ice, waiting ready to go. It's pointless trying to make a buffer partway through an extraction process. You're just wasting your time. You want everything to be, as I said, fresh, but not that fresh. You want to be also pre-cooled. And one thing is, in regards to the proteins, once you've extracted them, is to make sure that you store them appropriately. Don't freeze them in 1 ml aliquots and undergo multiple freeze stores cycles. All you're going to result in is your protein being degraded over a period of time. It's better to take smaller samples, let's say 20 microliters per aliquot, aliquoted and freeze them. You snap freeze them using liquid nitrogen and store them in the -80. And then take each aliquot for when you need it to run it on a gel, or when you want to do some further analysis, for example, a co-IP experiment. So the SDS-PAGE gel, again, I'm going to assume that everyone is aware of the various rigs that are out there and has a basic understanding of the procedures. This is more of a high-level understanding to try and just offer some advice in regards to the SDS-PAGE system. You know, one thing is to make sure that your SDS-PAGE rig is clean, whether it be a small or a large rig. It's important to keep it clean, because what you don't want is a situation where you have residual proteins left over from previous runs, and you actually then run that through your gel, through your fresh gel, as you're running the gel to resolve your current proteins of interest. And one thing is that it's really important to choose the optimal percentage of SDS-PAGE gel. For example, it's pointless running a 14% gel if you're looking for a protein that's migrating around the 170, 180, 200 kilodalton mark. I know some people that used to work on P300, which was a 300 kilodalton protein. So therefore, what I used to do as a scientist is I would, if I had a low molecular weight protein, sorry, a high molecular weight protein, let's say 170 kilodaltons, my choice would be to run a large rig and run it at 8% gel, use an 8% gel, and run it actually overnight. And this is sort of move on to the next point here, which is I found that by running a gel slowly and having a low percentage SDS-PAGE gel, that allowed me to make sure that the protein migrated through the gel, through the resolving layers quite nicely. And many a times I have benefited from this simple knowledge. Understanding exactly where your protein is in regards to its size allows you to engineer an optimal SDS-PAGE gel protocol and also a running performance. One thing is, and I've seen this happen before, is people who boil their samples, they put it in the SDS loading buffer, and what they realize that partway through the process is they haven't actually turned on their hot block. So you've got a hot block which is standing at room temperature, which you then have to wait for it to increase to 100 degrees Celsius. People will then take these samples which have been sitting there in SDS-PAGE slowly denaturing, and then add it to the hot block to complete the denaturing process. In my mind, if you do that, I would scrap those samples and go and get fresh samples from the -80 or prepare fresh samples. I found, based upon my own experience and my own troubleshooting, that if you put samples in SDS-PAGE jet, in SDS loading buffer, and then don't boil it immediately, it can actually cause the proteins to degrade significantly. I can't quite understand why that should be the case, but from my own experience, I've proven this time after time. One thing is that's critical is to run a reasonable amount of protein. Do not overload the protein, do not overload the system. For me, if I was using a mini gel system, I would run about 20 micrograms of protein. If I was running using a large SDS-PAGE rig, I would run at maximum somewhere between 40 to 80 micrograms of total protein. So this would be cellular lysate, it might be cytoplasmic lysate. It all depends, as I said, on the design process, which I highlighted to you earlier. But one thing is that it is important to measure your protein concentration before you add it to the gel. You want to make sure that you're adding exactly what you perceive it to be. There's plenty of systems that are out there for measuring protein concentration. Just make sure that you have it measured and you know accurately how much you're loading on the gel. It's one of the, again, it's a very important point, especially if you want to load equal lysates to show some kind of overexpression or underexpression. So you want to make sure that your total cell, your total protein that you've loaded on the gel is optimal. So should I say that is equal amongst the various different lanes. And also in regards to that, if you are sort of shown post-translational modification, one example is with phosphorylation, is you see an actual gel shift in regards to the protein itself migrating a bit higher in the gel the more it's phosphorylated. Now this is very difficult to prove if you have a gel which is actually what I term as a smiling gel, where it's lower in the middle than at the two ends of the gel. So again, by having everything balanced, by running a gel slowly and making sure that you have equal amount of proteins in each lane, this therefore prevents this smiling process or this artificial smiling. Again, this is something to do with unequal migration of the proteins through the gels. And although this may sound obvious to you, make sure the SDS-PAGE is connected correctly to the supply. I've done it before. I've been in a hurry to get out of the lab. I'm running a gel overnight, so what I do is I connect the negative to the wrong connection and the positive to the wrong connection and what happens is my gel, I can't understand why my gel hasn't worked in the morning, when all I've done is I've ran the system backwards, so that instead of the proteins migrating through the gel, they've just sort of left the lanes and diffused through this into the top layer of the tank. Again, just make sure you connect the positive to the correct connection point. And as I said earlier, depending on the situations that you use, you need to optimize the running conditions. As I said, one of my major things is I struggled with a protein for many years, which migrated about 170 kilodaltons. I found that by running it overnight in the cold room at an extremely low voltage, allowed it to migrate nicely through the gel. I don't know why this was. I think it was to do with, I can only sort of hypothesize it was to do with the hydrophobicity of the protein itself. But again, if I tried running this through a mini gel system for 1 hour, I had no success, even if I ran it at a lower voltage. So again, just to reassure yourselves, it's key that you optimize the running conditions for the protein of your choice. And again, a lot of this can be optimized through spending your time at the desk on looking at the internet and reading the research papers to understand what other people have done. You know, generally when I was in the lab, I had many people contacting me from other laboratories who were having the same issues with this exact protein and asking how I managed to optimize conditions. And generally with the more scientific community, people are more than happy to offer their hints and tips, and this has been very successful in generating more literature for publication. Or not, sorry. Yeah. Again, the Western blot transfer, this can be a very difficult step to optimize. Again, needs a lot of time and a lot of effort to try and to get it right. But if you're smart and you follow a few guidelines, I'm sure you'll not have struggle for too long. Again, one of the things, can't stress it enough, ensure your transfer apparatus is clean. I've had this situation where I've ended up with a blot which is blotchy, and generally that's been a result of the transfer residual protein being left over from previous transfers, and sure enough, it's contaminated my membrane when I was probing it. One thing to take into consideration is always wear gloves when you're handling proteins, whether it had been in the previous steps, but most importantly at this point in time. And one thing is, don't wipe your face with your gloves, because all you're going to do is you'll put proteins from your face onto your gloves, which you'll then transfer to your membrane if you handle the membrane using your gloves, which at the same time. And again, here we have an interesting option is whether you opt for the wet versus the dry transfer. And again, this is a result of, depends on the protein of choice. I refer back to my 170 kilodalton protein, which I spent a lot of time struggling with. What I did was, in the end, if I used a quick dry transfer system, I had no success whatsoever. This is why I had to, after spending the night running the gel slowly, I would then come in the morning, I would then set up a wet transfer system, and what I would do is I would actually run it at a low voltage, and I would use a wet transfer system, and I would run that throughout the day, at least 8 hours. And this ensured that I got efficient transfer of my large protein from the gel, and that it migrated onto the membrane itself. Again, as I said, this depends on your choice of your choice of protein. Now the choice for me was the Immobilon P, which is supplied by Millipore. I really had a lot of success with this membrane, with this system. I never transferred anything else throughout my Western blot career, or through my scientific career, should I say. And I can only remark on this. It was a great system for me. I never had any problems, and the only thing that I found was, once I activated with the methanol, I would recommend not allowing the membrane to dry out. It's very important because once it's activated, it's important to put it into the membrane the transfer solution, and therefore you want to make sure that you do not allow this protein to dry out, allow the membrane to dry out while you're doing other things in regards to, let's say, preparing the gel for the transfer process. One thing is that I used to actually have two membranes, one under the other, and the reason I did this was for smaller proteins. I could actually see if I looked on the second membrane, the transfer of the smaller proteins. Sometimes this was critical if I was working on proteins that were only, let's say, between 10 to 20 kilodaltons, but sometimes it was just useful to make sure that I hadn't transferred all my proteins from my first membrane through to my second membrane. As I said earlier, it is important with the Immobilon P to stop it drying out. And one thing that's worth taking into consideration is I used to equilibrate it in transfer buffer with shaking for about 5 minutes before I'd actually then add it to the transfer system. And what I used to do is I would set up my transfer process as highlighted below, which would include the filter paper, the membrane, and the gel. And what I would do is I would have 3 layers of filter paper, followed by the membrane itself, then the gel, and then on top of that I would have 3 layers of filter paper. Again, people sometimes have only 2 layers of filter paper. For me, filter paper is dirt cheap. I found it worked very well. It created a nice sandwich between the two electricity points, and therefore by having a nice sandwich, it ensured that I had a good connections between the 2 plates. One thing is that I commonly saw with people is they would actually they would not roll out any trapped bubbles within the system itself. So what you're going to have is a situation you would have is that there would be a bubble between the membrane and the gel itself, and obviously you're not going to get efficient transfer because you're not going to have the membrane in contact with the gel. So, just for the sake of which is a 20-second process, is to just take maybe an old biro or pen, take out and roll the biro over the system, over the gels itself, and over the sandwich itself. And what this allows is just allows you to remove any of the bubbles that are trapped within the sandwich, the transfer sandwich. And then all I can say is you try assemble the transfer apparatus following the manufacturer's protocol. Make sure you connect the transfer power supply as recommended. Just making sure that you connect the positive to the positive and the negative to the negative. You don't want your proteins migrating into the filter paper rather than migrating onto the gel. And as I said, I either opted for overnight wet transfer at 40 volts, or sometimes I did it for 8 hours throughout the day, depending on how critical or how fast I needed the result itself. And then you come in the next day, you're at the end of the evening, and you want to confirm that the transfer has been successful. One of my greatest discoveries, that I discovered, was a technical tip. Was I used to use Ponce OS to stain the membrane, which allows you to clearly see that the proteins have transferred onto the membrane itself. But I would also stain the gel with Kamasi blue, the one that after the transfer, just to make sure that there was no residual proteins left over in the gel. Sometimes you would have small amounts for the higher molecular weight proteins, but again, you're going to have to accept that that's one of the limitations of the system. You're not going to always get 100% transfer. And one thing is with the Ponce OS is before you proceed to the blocking stage, where you're going to block your membrane, what I would strongly recommend is that you make sure that all the Ponce OS is removed, and you simply do that is by washing the membrane in water, plenty of copious amounts of water added to the gel, and leave it shaken on a shaker for 5 minutes. And I would recommend doing three 5-minute wash steps, just to make sure that you got rid of all the residual ponce, because what you don't want it doing is affecting your transfer process, or your probing process further down the line. Blocking, again, there's some technical hints and tips which can be very useful here. One thing is, never allow your membrane to dry out. As I said, once you've taken it, you've done your ponce stain and you've washed away the residual ponce, get it in. Don't allow it to dry out. Keep it in the water, or get it into the transfer buffer as quickly as possible. But if you leave it on the tables, just dehydrating, or it's not going to be beneficial to your downstream steps. Now comes the buffer of choice. People commonly use PBS versus TBS. Well, I found that I had more success with tris buffer saline more than PBS. Again, this could have been just a preferential. I found that I could take TBS and with a Western blot and a new antibody that I've never tried, create an optimal blot within about three rounds of optimization. So, for me, TBS or tris buffer saline became the buffer of choice. Now, in regards to your proteins that you add to the blocking buffer, you can either opt for non-dry fat milk or BSA. Again, this has to become based. You have to do a lot of optimizations to understand what's going to work best for your antibody, and therefore for your protein, for your target protein. My preferred choice was 5% milk, non-fat dry milk. I did try BSA several times, and based upon my experience of 3% BSA, unless I had a weak binding monoclonal antibody, generally I would end up with high background with BSA, whereas with non-fat dried milk, it seemed to effectively block all the non-specific binding opportunities on the membrane, and therefore that resulted in a nice Western blot, or a nice film being produced from the Western blot itself. And I cannot stress this enough, please make sure that all your buffers are dissolved properly. I've seen it time after time where people will be in such a hurry to get on with their experiment. They'll add their 5% milk. They'll not allow the system to, to allow the stirrer to dissolve the 5% milk efficiently, so effectively, should I say. And what they'll result in is a mixed solution of powder, which is some of it is in solution and some of it's not in solution. Spend a bit of time, 10 minutes prior to starting the blocking step, to allow a bit of time for your milk or BSA to dissolve efficiently. And one thing is, sometimes people probe their membranes overnight at 4 degrees C, which makes sense. You don't want your milk going off. But what I could never understand is the same individuals would store their blocking solution at room temperature. Now, I used to do my Western blots in San Francisco, so room temperature in San Francisco could vary from 20 degrees during the day to sometimes in the summer it would be 27, 28 degrees. We would all go home in the evening, and people would use this gone off milk the next day, come in and find their milk gone off, but they would continue with the Western blot using the same blocking solution. This is not a good practice. I would recommend taking your blocking solution and putting it in the fridge. My recommended blocking time I would say is 1 hour at room temperature. And I would make sure that don't be tight in regards to your blocking solution. All it is salts and some non-fat milk. It's not expensive reagents, but your time is more precious to you. So why not spend a bit of time using for a 10 by 10 centimeter blot, I would use some for the whole of the system, including primaries, secondaries, wash steps and blocking. I would use somewhere between 1 to 2 liters of blocking solution, and also in the wash steps and everything. So that's how much I would I would use to make sure I ended up generating some really nice blots. So as I said, don't be tight in regards to your blocking solution. Make sure there's plenty to cover the membrane and plenty to allow for effective blocking. It's important to agitate gently using a rocking platform. It's pointless just having it sitting on the desk. And the other thing is, please remember to use gloves and tweezers. Don't wipe your hands all over your face and then start handling the membrane with your gloves. You know, as I said, all you're doing is you're going to end up transferring contaminated proteins from your face to the membrane itself, and therefore you're going to end up with a high background when you develop the Western blot later on. So primary antibody, you know, it can be very difficult to try and optimize which conditions are best for your primary antibody. You've got various different factors which come into play, whether it be a monoclonal or a polyclonal, whether it be a high affinity binder versus a low affinity binder. Again, it's going to vary on a case-by-case basis. But this is how I always optimize my antibodies. I first of all started with a dilution of 1 in 1,000, 1 in 1,000. The buff optimization, as I said, PBS, TBS, depends on personal choice. I preferred PBS, and I would start off with 5% milk. If I had no signal, I would then take it down to 1% milk. And generally, even for monoclonal antibodies, which were low expressors, which were low affinity binders, that would be sufficient for me to be able to detect the signal. Again, tween plus or minus, I normally use tween at around 0.1%. But for me, I preferred using TBS solution with tween at 5% milk as a starting point. And as I says, incubation period depends on your blot. I had some blots I could have an incubation period as short as 1 hour, but for others which would take more, which would include an overnight incubation at 4 degrees Celsius. But one thing that's worth taking into consideration is, you know, if you're going to incubate it for 1 hour, please keep it at room temperature. All you're going to do is reduce your binding kinetics if you keep it at 4 degrees overnight. The reason I used to keep it at 4 degrees overnight was to allow me to prevent degradation of the antibody, or to prevent instability of the antibody during the incubation step. So as I said, onto the washing steps. You know, you've invested a lot of time now in getting your Western blot to this step, and this is one of the many steps that I see people fail, is they actually they're very tight in regards to the number of times they'll do wash steps, and also in regards to their wash buffer. As I said, large volume of block and buffer during the wash steps. Normally for 1 centimeter square of membrane, I would add at least 1 ml of blocking solution. You know, it's important to do the agitation at room temperature, and I would always do 6 by 10 minute, 6 washes at 10-minute intervals. So I would make sure that I would change the wash solution, or which I've termed here as block and buffer. I would change it 6 times every 10 minutes. You know, you cannot wash, you can't take washing to extreme, but I think 6 by 10 minutes is a very sort of hits the nail on the head. It's a good sort of starting point. And now on to the secondary antibody. Again, this is going to be dependent on your choice of how you're going to sort of analyze your Western blot further at the end of the process, whether you're going to use ECL and film, or whether it's going to be a fluorescent label and you're going to use some kind of gel documentation system. But one thing that's worth it with noting is, with these type of antibodies, the secondary antibodies, they have high affinities for the primary antibodies. Therefore, you don't need to have a 1 in 1,000 starting dilution. My recommendation is 1 in 10,000. Sometimes I would have situations where the blotting the detection signal would be so strong and it would result in such a high background that I was able to take my secondary dilution down to 1 in 100,000. As I said, just to make sure, you have to understand which secondary antibody you want. Make sure that it's, if you're going to use HRP, that you're going to use it for ACL detection. If you're going to use fluorescent label, that you're going to use this for detection in some kind of gel documentation system. And generally, I would recommend, once you've added your secondary antibody, is incubating at room temperature for 1 hour. Same, and going back to the wash steps, it's the same advice that I suggested in regards to the primary antibody. Again, don't be tight in regards to your block and buffer. It costs peanuts to put it together. Your time's more precious. Spend more time focusing on, invest your time wisely in trying to get your blot right this time, so that therefore spend more time in regards to your wash steps. And then we come to the detection steps. So how you want to detect your protein. Let's say if you use the secondary HRP antibody, remove the excess wash buffer using a paper towel. What I used to do is just touch the side of the corners of the membrane, not the actual membrane. It's not the face surface of the membrane, but just the corners, to allow the excess liquid to drip off. And if I was using HRP labeled secondary antibodies, I would therefore then wrap it in I would expose it to ACL, drip off excess film, drip off excess ACL, and then I would expose it, wrap it in cling film, and then I would expose it to the film. Sometimes the exposure time would be a matter of milliseconds versus an hour or two, sometimes overnight. Again, it all depends on how strong your Western blot is in regards to its signal, and that involves the primary and the secondary. Obviously fluorescently labeled antibodies are very powerful. They allow for some really nice techniques, and I'll go through that later on throughout the talk. But it's important to use the appropriate machine. But also what's one tip is, when you're doing all your probing steps, especially with the secondary antibody, also with the wash steps, due to photo bleaching, make sure you keep your antibody and the membrane in the dock once the secondary has been incubated with the membrane itself. And that kind of is where I want to sort of discuss or end the discussion point in regards to my hints and tips in regards to Western blotting. And what I've done is, all these handy tips that I've gone throughout the talk, is I put it into a nice 2-page document that you can contact one of the Innova team to allow you to be able to obtain this in a PDF format. And what this will allow you to do is, as you're going through your Western blot procedures, you can nicely tick each individual box itself to say that you've done these steps. And then what this is going to do is allow you to at least provide a framework to understanding what might have went wrong. Let's say if you get a blot which has such a high background. Let's take an example. You have a high background blot. You've done your desk research, which says that you know where the protein's expressed. You've done the protein extraction. You've ran the correct percentage gel, and on top of that, you've confirmed that the transfer has been successful. Your only sort of variables at that point is your primary, your secondary, and your blocking solution. So you're really starting to nail down the variables in your experiment. And then what you can benefit from at this point is, you can then say, okay, I've ended up with a high blot, a high background blot. This could be the result of the too much secondary antibody, not enough washes, or possibly that I need to decrease the amount of primary and secondary antibodies, increase the amount of blocking solution, as in the percentage blocking solution, and also increase the amount of wash steps, so you're washing away non-specific binding. So hopefully, I hope this has been a benefit to you, and it allows you to really understand, in regards to someone who's spent many years at the bench trying to understand how to optimize a Western blot, this will provide a framework for you to be able to, like I did in the end, to be able to take any antibody and quickly optimize the conditions for probing and for a Western blot experiment. You know, I went from way back in my early PhDs to spending 2 or 3 months over a period of time optimizing a blot to actually being able to optimize an antibody towards the end of my postdoc within about 2 or 3 days, and that's by having this framework in place. That's how I really benefited from it. I saved myself a lot of time. And you know, and this is what I said, by having this framework in, you can build your engine, you can have checkpoints all through the system, and then you can turn the key to make sure that it starts at the end of the day after you've put in all the effort. And again, here we are at the start of the process. You've ended up with a blank blot. You've applied your framework. You've taken on some more extra work. You've resulted in now the signal being present, but still some non-specific binding. Some more troubleshooting, and what you end up here is all of us jumping for joy because you end up with a beautiful and perfect blot as outlined here. If you have any further questions, as I said, Claudina at the end we'll have a question and answer session, and Claudina will chair this, and I'll be more than happy to answer your questions. So I just wanted to share, so for the second half of the webinar, I just wanted to share some sort of advancements in regards to Western blotting and how the technology, I've observed the changes in the technology over the years. You know, in the past, this was very much the standard where people would use chemiluminescence. You would get a substrate, you would have an enzyme present on your secondary antibody, in which would emit, there would be some, which would result in the substrate converted into light, which could then be detected by exposure to film. Whereas here, what now people are doing is they're taking fluorescently labeled secondary antibodies, and through excitation, and what's happening is the fluorescent label has been excited, which is resulting in the emission of the signal, which can be detected on the appropriate gel documentation system. And you know, there's really great benefits to being able to detect fluorescent proteins of fluorescent Western blotting. And here's an example. Here is there's two where the researcher has probed the same membrane simultaneously with 2 different antibodies, each raising different species, so they're not going to encounter cross-species reactivity. And what they were able to do is they were able to use these 2 images and combine it and overlay the images to create a pseudo image, which reflected in here in red is one antibody, and the other in green is the other antibody. And as you can quite nicely see, this antibody here contains a mixture, or this band here contains a mixture of both proteins migrating at the same size. And here's another experiment where the researcher has run the gel in parallel. They probed one of the gels with beta-lactam here which is in green, and with the other gel they actually, instead of transferring it, they stained it with Kamasi stain, and then what they did was they then visualized this in a gel documentation system, and they were able to overlay the 2 images once it was all pulled together to actually show that even though they got equal protein load in here, there was variation in regards to the beta-lactam expression. Again, this sort of shows you that sometimes when you're trying to show equal protein loading, it's sometimes necessary to have other controls, not just being beta-lactam. So one thing is, you know, is the powerful of this technology is through fluorescent Western blotting, you can actually probe the same membrane to detect 2 different proteins at the same time. A really powerful offering. The downside is in regards to a situation here where if you have secondary antibodies, which are used against the same species. For example, here we have several different proteins which you want to detect on your membrane, all raised in the same species. And what you're trying to do is you would, unfortunately, because you use a secondary antibody, you can't use secondary antibodies to detect the different proteins. It's all just going to show up as the same signal, because unfortunately you're going to get cross-species reactivity, even though you've engineered the antibodies to have different signals, different fluorescent labels. Unfortunately, what's going to happen is you're just going to get cross-species reactivity, or the secondary antibodies recognizing the same primary antibodies. Now, there is benefit in regards to this, of getting over this images. You can actually eliminate the need for secondary antibodies by using direct detection. And this is where the primary antibodies, regardless of species, have been labeled with a fluorescent dye or fluorescent protein. So here we have, even though they've all been raised in the same species, here we have them all labeled with three different labels. Therefore, by using the appropriate gel documentation system, excuse me, and the Western blot and Western blotting technique, you can detect the different proteins simultaneously and probe them at the same time. Excuse me, I'm just going to take a drink. Thank you. And again, there are benefits with this indirect versus direct detection. And here's an example of here in the past where I'd actually performed an immunoprecipitation experiment where I pulled down an antibody called beta-catenin. And then what I had to do was, because at the time there wasn't a lot of beta-catenin antibodies available, so I couldn't pull down with one beta-catenin antibody raised in a species and then reprobe with a different beta-catenin antibody raised in a different species. I had to actually just pull down with the beta-catenin antibody, and then I had to reprobe with the same antibody with cross-react, and then use the same secondary antibody. And obviously, because you have heavy and light chain present within the system, we end up with this IgG band present in the Western blot itself. And as you can see, because there's so much heavy chain there, what's resulting is I'm getting a lot of non-specific binding, especially where the beta-catenin antibody, the beta-catenin protein is migrating. It's just a smear, unfortunately. And this is actual just input. This just shows you where the beta-catenin antibody, the beta-catenin protein itself migrates through the gel. So again, I was lucky because my beta-catenin protein did not migrate at the same level as the heavy and light chain. So you can imagine if it did, I would never have been able to show whether the beta-catenin protein was present in the sample due to the high signal from the heavy chain. Now this has been overcome by the scientists here, and what they did was they pulled down an antibody. They pulled down a protein called, we'll just call it E here. They pulled down this protein called protein E, and what they found was, through a silver staining experiment, that the protein E itself migrated just above the heavy chain. So here we have it, the heavy chain, here's the light chain, and here's another protein of interest, which is protein M. But what they did was they took the same antibody here, this H5, and they labeled it directly using a HRP labeling kit, and through that they were able to prevent the need of the secondary antibody, and therefore they didn't end up with this heavy chain signal that I experienced in this situation. So here's the situation. They've got this beautiful Western blot, which clearly shows the presence of the specific antibody of interest, but not any other lanes. Now, as I said, if they probed back using a secondary antibody, based upon this experience here, they would have ended up with a lot of with the heavy chain signal being extremely strong, which would have most likely masks the true signal of the E protein itself. So as I said, there's many advantages of indirect versus direct labeling. Essentially, it allows you to eliminate the need of secondary antibodies, therefore you don't have to do those additional wash steps, the additional incubation step of the secondary antibodies. More importantly, you can probe the same antibodies probe the same membrane using antibodies or primary antibodies from the same species, and therefore you won't get cross-species reactivity as you do in this situation here where you have to use the secondary antibodies. And more importantly, it allows you to probe the same membrane several times. So we obviously are aware of the advantages of direct labeling, as just highlighted here. But what's worth noting is there are major problems with labeled antibodies, with trying to identify labeled antibodies. Basically, there's a lack of availability out there. Commercial suppliers have done a very good job of providing labeled antibodies, but unfortunately there might be only two researchers in the world that want a specific antibody labeled with a specific protein or dye. Now the downside is that's not commercially viable for the wholesalers to provide that antibody. Therefore, they're never going to cater to meet that target needs. There are kits, or there is a conventional approach where you can label the antibody yourself using traditional methodology, but there are downsides. You do need a lot of understanding of the basics of the chemistry, whether it's going to be in an amine group you're going to target, a thiol group, the type of chemistry, the type of chemicals you're going to need to drive this the labeling of your antibody. And it also can take up a significant amount of time. So not only do you have to have the expertise, you have to have the time. A traditional labeling experiment can take 1 to 2 days. It can involve a lot of column separation steps. You also can need a lot of starting material. Sometimes you can need somewhere between 1 to 5 mg of material, which can be a bit of a costly experiment, especially if you want to do a one-off experiment just to see if your protein is there or present within the sample. You don't want to be investing in 5 milligrams of antibody to be labeled. Also through the separation steps, you have to invest. You have a lot of wastage because there will be some wastage of material. You may only recover somewhere between 50 to 70% of your material. Therefore, you're going to end up losing money in that respect as well. So to overcome this, we as a company, Innova Biosciences, developed our Lightning Link technology, which we branded as the world's easiest to use antibody labeling kits. And really, all it is, it's a 30 seconds hands-on time. All you have to do is take your purified antibody, add it to the freeze-dried powder. Now this contains your label of interest, this contains all the chemistry, and then what transpires is the labeling reaction is done within either depending on the format of the kit, is done within 15 minutes, or it's done within a 3-hour process. After this, all you have to do is add your quencher to stop the labeling reaction, and then therefore that's your antibody labeled and ready for its Western application. So you don't have to do any separation steps. There's no need for any expertise in regards to the chemistry driving the technology. It's just a very simple and nice technology to use. We have over 50 labels, including enzymes, fluorescent proteins, dyes, tandems, biotin, and streptavidin. And therefore that means whether you're doing Western blot using ACL detection, or you're doing Western blot using fluorescent detection, or using a fluorescent system, should I say, you're not going to have a shortage of options of dyes to label your proteins with. So just going back to reiterate the points, chemistry expertise is not required. You end up with 100% antibody recovery as there's no separation or dialysis steps. Now, more importantly, our pack ranges from 10 micrograms, 100 micrograms up to 5 milligrams. And what's encouraging or what's beneficial to our customers is it's a covalent conjugation which is formed between the antibody and the labeling itself. Therefore, it ensures long-term stability. We have 12 ongoing stability trials, and I think the latest we've done is up to 18 months, which demonstrates as long as the conjugate is stored at the recommended guidelines, let's say 4 degrees C, if it's a fluorescent dye in the dark, the performance of the stored conjugate will be as good as the fresh conjugate. And therefore it allows you to create a conjugate, and if you have 100 micrograms, you use 10 micrograms in a Western blot, you still have the other 90 micrograms of conjugate for use further down in the future, whether that be in a couple of months or in a year's time. And we have two ranges of Lightning Link. We have a Lightning Link Standard, which is a 3-hour incubation, and Lightning Link Rapid, which is a 15-minute incubation. Again, there's lots of information on the website, or we can answer some questions in regards to Lightning Link in the FAQ section. Sorry, in the answer and questions section later on. And as I said, just to reiterate, these are all the various kits that we have available on the website. The enzymes, the fluorescent proteins, the biotin streptavidin, the tandems, and our fluorescent proteins, our fluorescent dyes, ranging from the UV to the far infrared. So just in closing, I wanted to discuss one more point with you, and we commonly encounter this, or questions in regard to this, and it's to do with the secondary amplification hypothesis. And if I just go back for a moment, what people perceive is if we have a situation where we're eliminating secondary antibodies, we're not going to have this amplification step that people perceive occurs when you don't have secondary antibodies present. Now, we've done this for a panel of antibodies, a panel of 4 antibodies, and this was independent of our scientists. This was an outsourced project where we took the same antibody and we took this protein, which is a quinine reductase 1, ran it on a gel, labeled the same antibody, and directly with HRP or indirectly using secondary HRP labeled antibodies. And if you note here, the directly labeled antibody is diluted significantly more than the indirectly labeled antibody. And if you observe the intensity of the signal, you'll observe that the primary antibody, which is directly labeled, is equivalent to what it is in the indirectly labeled antibody, indirectly secondary antibody. Now, we can't say this is going to be across the board. We've done this for 4 proteins, and we found that this is the case. So therefore, just to offer reassurance, don't necessarily assume that just because you're eliminating secondaries that the signal will not be as strong as if you were using the secondary itself. It may not be the case. And in summary, I just wanted to discuss what we've talked about in the webinar. I hope you found the troubleshooting framework useful for you in regards to troubleshooting your own experiments in the future. We discussed the indirect versus direct detection methods and the benefits of direct detection, the problems of sourcing labeled antibodies or labeling antibodies using standard methodology. We discussed the Lightning Link technology available from Innova Biosciences, and we briefly touched upon the secondary amplification hypothesis, which I hope will reassure you. As I said, if you eliminate secondaries, you're not necessarily eliminating the sensitivity of your system. And with that, I'm going to hand everything back to Claudina. Claudina, thank you.
Great. Thank you very much, Brian. So thank you very much for joining us today. We hope that you enjoyed it, and we hope that you'll be able to join us for the next webinar in December.